But yeah, thank you for coming, everyone. Welcome to our Integrated Pest Management Academic Seminar Series, which is designed to increase awareness of new research and techniques that advance IPM and its adoption in all types of pest management settings. These seminars are typically once a month during the academic year, and you can find these events as well as some of our other events like our What's Bugging You First Friday series on our website, which I'll link to in the chat in a minute. By the way, this is also streaming via Zoom, and there's maybe 20 or so people coming on there. So now it's my pleasure to introduce Kiran Gadhave, an assistant professor of entomology at Texas A&M AgriLife Research and the Texas A&M Department of Entomology. His academic journey spans 3 continents, a master's in entomology from Tamil Nadu Agricultural University in India as a junior research fellow, a second master's in plant breeding and genetics from Cornell as a Tata fellow, and a doctorate in biological sciences from the University of London as a college overseas scholar. Now at Texas A&M, he leads an interdisciplinary research program at the intersection of insect vector biology and molecular biology and plant virology. So let's welcome back to Cornell our speaker, Kiran Gadhave.
Well, thank you so much for that wonderful introduction. It's great to be here after 17 long years. A lot of nostalgia. But yeah, and thank you for that kind introduction, Brian. Appreciate it. So plant virus vector systems are often described as interactions, and I don't think that's the right word. They're really battlegrounds. And in that battleground, tomato spotted wilt virus, which is going to be the antagonist I'm going to talk about a lot today, has been winning for a long time. So this virus infects more than 1,000 plant species. It's transmitted by several species of thrips. And for decades, our go-to or reliable strategy to fight this virus has been to use single gene resistant cultivars. And that worked for a while, but what's something fundamentally changing in the last couple of decades, and that is the evolution of resistance-breaking strains, which are spreading globally. A lot of them remain unreported. And most importantly, it's undermining the most reliable defense that we had, which is the use of resistant cultivars. So now the question is no longer how do we control this virus, but the real question is how do we stay ahead of the system that's constantly evolving against us? And that's what I'm going to talk about today. So I'd like to give you a brief overview of my academic journey before I begin. It all started when I was born, which helps. So I did my undergrad and master's from the first master's anyways, from India. The second master's from here, Cornell University, it was actually in plant breeding and genetics. And then I did my PhD from University of London in the UK. Then a couple of postdocs later, I ended up in my current position at Texas A&M University, where I lead interdisciplinary insect vector virus interactions, or rather system program. And I've been very fortunate to be able to travel to different places as business or scientific events. So I took immersive learning at Cornell very seriously. Yeah, I don't even like beer, so. But this is the place where I started asking right questions. So our story is going to be in 5 parts. First, I'm going to talk about problem that we are facing, or rather multiple problems, the mechanisms underlying that, detection, control, and prediction. So the goal is to move from biological chaos to something that's more predictive and controllable. Tomato spotted wilt virus, as I mentioned, is one of the most devastating plant viruses or diseases in the world. It infects more than 1,000 plant species. It's transmitted by several species of thrips. And the most challenging part of this system as a whole is that it's able to evolve not only in plants, but also in the vector. The most reliable strategy has been the use of resistant genes. That resistant came in the form of SW5B. Oh, let me use the pointers. Okay. In tomato and TSW in pepper. But the strong selection pressure by resistant cultivar has led to evolution of resistance breaking strains, as I mentioned, reported all over the world. And in the US, they are likely to be present way more than they have been reported, but so far reported in Hawaii, California, North Carolina, Louisiana, and Texas in the US. So this is resistance breaking in action. You can see the field images where you can see characteristic tomato spotted wilt virus symptoms in resistant cultivars. And what should be resistant basically is no longer holding, and that's a big problem. Let's quickly look at the TSW genome organization. This is a RNA genome. It has 3 segments named after their sizes. L encodes RNA-dependent RNA polymerase, M encodes movement protein and as well as glycoproteins, and S encodes silencing suppressor as well as nucleoprotein. It's important to understand this genome a little bit better so that we can understand what's going on in terms of resistance breaking at the molecular level. So the first big question we asked is what's driving resistance breakdown at molecular level? Our 2023 field season started with 6 different isolates. I'm going to use those, I might use those terms interchangeably, the isolate and strain. That's because in the initial stages, we worked with isolates, and then once they proved to be strains, that's where I use that term more in a more robust way. So we had 6 isolates at this point. 5 of them were resistance breaking, 1 was non-resistance breaking, 4 of them came from tomato, 2 came from pepper. And so if you look at the percent nucleotide identity here, most of them look pretty similar, whereas except for the TOM-CA, which was a bit different compared to the rest of the isolates that we had. And so we map these isolates phylogenetically, and what we saw on the basis of nucleotide sequences, the entire entire genome as well as L protein, there was a clustering of these isolates, especially the tomato ones, together. With the S segment, based on nucleotide sequences, again, M and NSM, the clustering was different. We had 3 isolates that grouped together, whereas the TOM-CA, the one that I mentioned earlier, was divergent, actually clustered together with TOM-MX isolate, which was from a different group. And then in pepper, on the basis of S segment, we saw the grouping of these 2 isolates, the one resistance breaking and one non-resistance breaking together. So what this basically tells us is that there is a selection pressure being applied on the entire genome of tomato spotted wilt virus. Okay. Now, let's look at the positive selection sites. This table shows the statistically significant positive selection sites, 2 groups, tomato and pepper. We had several different proteins of tomato spotted wilt virus. These are the codon positions which were statistically significant. And I would like to bring your attention to these red amino acid substitutions, which were unique to our study isolates, and we are studying them further. What was most striking was this NSS 52H substitution at 462 amino acid position, which was the only point mutation that's unique to our isolate, as well as it was showing the strong positive selection. And so this was our kind of a candidate for resistance breaking in pepper, especially. Now, if you're interested in the full story about full-length characterization, full-length sequencing and characterization of these strains, this is the paper we recently published in collaboration with Dr. Srinivasan's lab at University of Georgia. Now, coming to 2025 isolates, we had 3 of them, and what we were interested in seeing is whether the resistance breaking mutations that we've been seeing in the earlier isolates are also present in these 3 isolates. And that's why we extensively molecularly characterized them as well as studied the phenotype. The first isolate came from Bushland, Texas. This is more north or Texas Panhandle of Texas. The Uvalde isolate came from the south of Texas, and the College Station, or CS for short, came from more central south of Texas. So geographically representing various regions of Texas. Because when we collected, we did not know their resistance breaking or non-resistance breaking status. So one of the first things we did is we tried to understand the hypersensitive response of these isolates in resistant versus susceptible pepper. The form of resistance in pepper is TSW. And so what we did here is agro-infiltrated NSS protein in resistant as well as susceptible pepper. And then what came out to be most distinct of these isolates is HT pepper. HT stands for high tunnel, and it failed to produce hypersensitive response in resistant leaf, as you can see here, which was kind of the first sign that this was a resistance breaking phenotype or isolate. And then we took the HT-PEP RB, put it together with the Uvalde non-RB isolate, and then we tried to understand the phenotype better in both resistant as well as susceptible cultivar. Now, as you can expect, both RB and non-RB were able to produce infection in susceptible cultivar, which we used was King Arthur. And you can see here as well the whole plant showing the disease phenotype. Whereas in the resistant plant, the story was different. This is the resistant cultivar. We used Frodo, and this is at 40 days post-inoculation. And so, as you can expect, the resistance breaking isolate, which is the HT prep, was able to produce infection in these resistant plants. And then that was confirmed by the phenotype you can see on the leaves here, as well as the plants here. The second resistant line we used was actually the line in development in the Texas A&M breeding program. And then this isolate was also able to break resistance here as well, as it's confirmed by PCR on the gel image, and also the disease phenotype you can see here as well as here. Now, in both resistant cultivars, we did confirm that the TSW did express the resistant single gene resistance that being offered is definitely expressed prominently in resistance breaking as opposed to non-resistance breaking isolate. The next question we try to address is, now we know or established that the HT-PEP RB is able to break resistance in pepper, but is it also able to break resistance in SW5B tomato? Because tomato is a different form of resistance, also single gene, but different. We need notice 3 things. The first one is that this isolate was able to produce symptoms in susceptible tomato, which is this SW5B minus M82 cultivar. The second thing we notice is that it is able to produce hypersensitive response in resistant cultivar, which is the mountain merit here. As you can see, the prominent hypersensitive response here. But what goes in hand with that is the lack of symptoms in resistant cultivars in mountain merit. You can see here in this phenotype. So yes, the HT-Pep RB was resistance breaking in pepper, but not in tomato. And so it was not a dual, what we call a dual resistance breaking isolate. This is interesting. So we sequence the NSS, or the entire genome rather, but this is the amino acid sequence of NSS. And we compared the HT-Pep RB isolate with the other key resistance breaking and non-resistance breaking isolates that we had. And what we observed is 4 unique point mutations happening across the NSS in various regions. The first one at 12th amino acid position, the second one at 139th, 328, and 361 amino acid position. The next logical question is that does that changes the overall structure of NSS protein? And the answer to that, based as you can see in the image, it does not. Then what happens at the intermolecular levels between resistance breaking and non-resistance breaking? As you can see, there is some subtle or rather distinct differences happening at the intermolecular interactions, intermolecular level. And so what does that tell us? It tells us that even though there's no obvious major structural changes in the NSS, but we suspect that it would actually impact how NSS interacts with host proteins that are involved in RNA silencing or plant defense signaling. So that remains to be seen, but it's very interesting what's happening at the molecular level. And this basically, the whole section provides a mechanistic explanation of what's happening in terms of resistance breaking. Now, if RB strains differ genetically, can we detect them rapidly? That's the next question we try to address, because once resistance starts breaking down in the field, the timing becomes everything. Why are we always late? That's the question I have here. In systems like this, we are always reacting late. We detect virus when the symptoms appear, and then we start deploying intervention strategies when those symptoms become worse. And by the time, it's already late because the disease is spreading in the field, right? It's being transmitted, it's being spread in the field. In that sense, we are always late, not because we do not have the right tools, but we do not know how to use them, or we are not using them in a way that would let us anticipate what's coming. And this is really the motivation behind the rest of my talk, is how do we go from reacting to anticipating? Now, here's a potential solution in the form of Cas13a enzyme, which we use for detection of tomato spotted wilt virus, which is an RNA virus. And this is basically the guiding principle. We start with designing a guide RNA. In this case, it's going to be tomato spotted wilt virus target. We then have a mixture of Cas13a and guide RNA, which scans the sample and binds specifically to the viral RNA sequence that we designed, which then leads to the activation of Cas13a, and then which leads to the lysis of this target molecule, which releases the fluorophore that is already present in the sample, and you see this nice, beautiful glow, which tells us that the virus is present, or whatever we are putting in there is present. Now, what does that look like for tomato spotted wilt virus in action? You have the CRRNA sequence shown by purple here, and this is based on the NSM protein, one of the proteins of tomato spotted wilt virus. And then we have the conserved non-viral stem loop, which is shown by red here. what you basically start with is a crude RNA extract with the lysis buffer, which takes about 10 minutes. The ribonucleoprotein complex, which is this guide RNA and Cas13a, the incubation takes about 10 minutes after this, once you mix the RNA, and then all you have really is the cleavage buffer and then the reporter substrate. You incubate the cocktail for about 15 minutes at 37 degrees Celsius, and that's very much the room temperature, or a little bit more maybe. So you put those tubes in the handheld fluorometer, and what you will see is this. And when you see a glow like this, that tells us that the tomato spotted wilt virus is present. Really cool, actually, if you ask me. And this is our proof of concept, right? The beauty of this system is, first of all, this is well under 1 hour. So I'm not waiting for weeks for the results to get back. Second, there is no PCR machine here, or there's no $10,000 being invested, right? And also expertise required. It's really simple. Once you get a hang of it, it's easy. So the point is, it's very much field deployable. Now, the coolest part out of all of this is not whether we can detect the virus, but can we discriminate or distinguish one strain from the other? And yes, we can, because now we know the sequence of all the NSS amino acid mutations that I explained earlier, and how you can distinguish between resistance breaking and non-resistance breaking strains, or between one resistance breaking strain to the other. We just change that sequence. We would know what 4 or 5 different strains are going to be in the field, and we should be able to use this principle to be able to confidently distinguish between RB and non-RB strain. So this is our current work, and we are seeing some promising results. So, so far, 3 things we have seen is resistance breaking is mutation driven. These mutations are very much detectable. And we are able to overcome some of the devastating limitations, or the key limitations of the existing detection tools, and then the devastating impacts associated with that. And then we can also use the system to potentially differentiate between RB versus non-RB strains, which is really cool. Which means that this is no longer a biology problem, but it's rather a timing problem. Which brings us to the next section, which is detection is not enough. That's good. It's cool that we have the system, but it's not enough. So if we can detect early, the next key question is, can we intervene even earlier? That brings us to CYVAV. It's a citrus yellow vein associated vector that we try to develop it as a vector. But it's actually an independent mobile RNA, which is associated with the rare yellow vein disease of citrus. And this disease was reported back in 1950s or 60s. And the RNA associated with it was actually first reported and characterized by our group at UC Riverside. This was my previous position. And this RNA is so cool. This is basically it, right? That it was licensed by Silvec Biologics. It's a startup company. And the reason for that is it's got all these prominent attributes that makes it such a cool virus-induced gene silencing vector. The most important being, it's so small. This is the genome organization, and this is it. There's nothing else. This is basically has the 2 open reading frames. They code for RNA-dependent RNA polymerase, the enzyme that's required for viral replication, using the -1 ribosomal frame shift. And that's what makes it such a cool RNA delivery system. This work received a lot of worldwide media attention. Let's look at the virus-induced gene silencing real quick. You basically have the target gene fragment of thrips or tomato spotted wilt virus, which is inserted in the CYVV backbone in this case, which is then cloned into agrobacteria and then introduced into plants using what is called agroinfiltration. And the idea here is as the virus replicates, and the first key step in that is the formation of double-stranded RNA, which is detected by the host cellular machinery using dicer-like enzymes, and that degrades this double-stranded RNA into small interfering RNAs, and then that leads to the binding of these siRNAs into RISC or silencing complex, and that leads to the degradation of target mRNA. And we are basically, what we are degrading here is the thrips or tomato spotted wilt virus gene or protein. We selected 4 targets from thrips on the basis of the key roles they play in thrips developmental processes, everything from cytoskeleton development to immune signaling. And the targets were actin, tubulin, VAT-base, and toll-like receptor. And then with tomato spotted wilt virus, we had 5 targets: the RNA-dependent RNA polymerase, movement protein, glycoproteins. These are basically different proteins involved in essential viral functions. And this was the criteria that we used for target gene selection. With the thrips targets, one of the first things we were interested in knowing is whether they play any key roles in tomato spotted wilt virus transmission. And for that, we used the infected tomato spotted wilt virus infected versus non-infected thrips. So we started with the first instar larva, and then we allowed them to complete the life cycle on surface sterilized green beans. That's a common procedure we use in the lab. Now, we quantified the relative gene expression at the second instar, pupal, and adult stage. First things first, we needed to make sure everything is working great, and that starts with the constructs. We had 500 base pair CYVAV constructs with these targets in the backbone, and we wanted to make sure everything is working before agroinfiltration, which is what you can see here, but also after agroinfiltration, 20 days, 8 days post-infiltration, which you can see here. We wanted to confirm the replication of systemic movement of CYVAV at 30 days post-inoculation, which we can confirm with the negative strand DNA synthesis, and then obviously wanted to make sure that there's no horrible symptoms being produced by this vector backbone in tomato, which we did not see anything at 20 DPI. These are the regular checks we imply in our lab. Now, this is really cool. What is happening here is you have 4 target genes. We have relative expression on y-axis, and then we have healthy versus infected on x-axis at different stages. What you will notice and is consistent is the relative gene expression goes down consistently in second instar, here, everywhere. Whereas these genes are upregulated, or it goes up as an adult. And the plausible explanation for this is the evolutionary adaptation. Remember how virus also evolves in thrips, is that thrips are allowing, or the virus is allowing itself to establish infection earlier on, and so downregulating these defenses. And once the virus is established, then that leads to increased expression, and that leads to ultimately increased fitness of thrips as a result of that. And we have independently verified that it's in fact a well-known fact that the tomato spotted wilt virus enhances the fitness of thrips. Very cool. Now, this is a brief overview of the methods we followed to study the silencing, the CYVAV-mediated silencing of thrips target genes. CYVAV constructs, as I mentioned, we agroinfiltrated them in 3-week-old tomato plants, and then we allowed first instar larvae to infest tomato plants 10 days post agroinfiltration. Now, we sampled at 2 different stages. First one at 48 days after exposure of thrips to to CYVAV, and continuous exposure, basically 12 days of exposure till they become adults, which is 22 days post agroinfiltration. And now what we looked at is two things. The first thing is relative gene expression, obviously, but then we also wanted to see what's happening at the survival, with the survival. First, with the 48-hour exposure, what we saw was astonishing. 50 to 93% reduction in transcript levels of the target genes. As you can see here, this is 93%, which is pretty cool. Then with the continuous exposure, it's a similar story, 68 to 92% reduction or silencing. And this is unparalleled because this is unlike we have seen before with any other vector virus, or the VIX virus system, or reported that we know of in literature. That's all cool with the gene silencing, but what about survival of thrips? And we saw, and these are preliminary results, we saw that there was a significant mortality of thrips, especially with the actin as well as TLR targets. And just switching gears a bit, now we know that CYVA is working very nicely against thrips targets. The next goal was to see if it works against TSW as well. These are the 5 targets, as I mentioned earlier. They are targeting critical genes of tomato spotted wilt virus. And these are 2 controls. The protocol or the methods remain the same until agroinfiltration. In this case, it was Nicotiana benthamiana. And where we infested thrips, instead, we mechanically inoculated these plants with tomato spotted wilt virus. And what followed after that is understanding the relative gene expression at different time points, 10, 20, 30 DPI, and so on. Again, very cool stuff happening here. The first image you will see is the actual phenotype. And NSM, NSS, and RDRP has the healthiest phenotype, as you can see, compared to wild type and other treatments. And that goes hand in hand with the severity, disease severity on the scale of 0 to 5, where 0 is healthy and all green, and then 5 is dead or collapsed plants. And so the middle 3 bars here, you can see there's a lot of green here at 10 days post-inoculation, as well as at 20 days post-inoculation. So these treatments are clearly winning. We wanted to see, obviously, the virus accumulation at the same time, how much TSW is accumulating in these plants. And the story was consistent as far as NSS is concerned. As you can see, the virus expression, the virus accumulation is significantly lowered compared to wild type here. But then that was not the case with the NSM or RDRP here. But that was the case with the GN, but then GN is producing this weird phenotype that the plants are dying. So this is only 10 DPI results, so we'll get much clearer picture when we look at more time points in the future. But this is a fresh data. So, but it's, clearly the NSS is showing quite a bit of promise with this. So in a nutshell, CYVV is an excellent platform, unparalleled attributes. We are able to target a bunch of different genes of thrips as well as tomato spotted wilt virus. The efficacy is up to 93%, and we are able to see the reduction in TSW severity as well as accumulation. Now, where are we going with this work? We have short-term and long-term goals. In the short run, we want to see how this silencing impacts thrips fitness as well as transmission. And then in the long run, we want to see what's the best path for commercialization of this technology and how best it can be integrated with IPM practices. And this is the shift I want to leave you with. This is the final part. So if we can reduce transmission, can we predict outbreaks before they happen? And in other words, can we turn the reactive management, which we do now, into proactive decision making. It brings us to machine learning. What this is it's a fancy term, first of all, but it's pattern recognition at scale. What happens here is that you feed computer, in simple terms, a lot of different kind of data, environmental, biological in this case, and then it shows us patterns or shows us relationships that we were not able to see before. It's amazing what it can do, and I'll show you in a minute how. There are three powerful tree-based models in this. There is a random forest, which basically when you feed the data to it, it will generate multiple trees based on this data and vote for the best answer and give you the final prediction. The other one is the gradient boosting machine, which builds trees a bit differently. It's sequentially, and every tree that's built reduces the error and it improves step by step. The extreme gradient boosting is the version of GBM on steroids. It's much faster, it's much quicker, it's much more efficient. And why three models? Because not all data are same. As we know, biological data is often context specific, it's nonlinear, it's messy. And the reason we employ these different models is so that we, depending on what our goal is and what kind of data we are dealing with here. The question we try to address is, what are the biological and abiotic factors that impact thrips population dynamics? And these are the different parameters. As you can see, wind conditions, there is 4 different variations of that because wind conditions impact how thrips move or spread. Humidity, because it impacts thrip survival. Temperature, because variations of this impacts the thrips developmental time and many other processes. Parent population, because that's the starting point. It's a key variable. So we looked at 16 different parameters all at once using machine learning to find patterns, that to see if we find any new patterns, but then we miss anything with the regular classical linear regression based models. Now, to make it further complicated, this is where things get interesting. We're dealing with not one system, but 2 different systems. One is open fields, as you can see here in this image, and the other one is high tunnels. And these are next to each other, basically. This is the setup. These are the open fields and high tunnels, and this is where the weather station is located. We had 2 planting dates. Why not complicate things? And then we had 12 yellow sticky traps. That's how they look, deployed every week at each site. And so that makes it around 1,600 traps in total over 5 to 6 months. And if one looks like this, just imagine 1,600, and counting them, it takes hours and hours and hours. So this is the blood and sweat of undergrads that we cannot be grateful enough, you know. So what we did next is, obviously growers don't care about what the exact number of thrips are, but they care about risk threshold. So we categorize this data into 3 different categories: low, medium, and high. Low, where we had less than 25 thrips per trap, medium less than 100, and then high more than 100 traps. And one of the first things you will notice is that there is a nearly symmetrical distribution of these thrips in high tunnels, whereas they were more asymmetrical in the field. More than 2/3 of the traps collected from the field actually had 100 thrips. So that tells you something. Now, how we built model is we use 75% of observations as a training data and 25% as a test data. Now, the performance part, and this is where it gets really interesting, is how these models perform. In high tunnels, XGBoost performed the best with 85% accuracy and lowest errors. In open fields, random forest performed the best with 88% accuracy and lowest errors. So the key takeaway here is that these are not just statistical differences, but what this tells us that these are two fundamentally different microclimates we are dealing with. And also these are potentially different micro ecosystems that thrips encounter as they build their populations. This is very cool. And the next big thing we observed is that the top three variables that were most consequential in terms of thrips population buildup were parent population, which was common across both. No surprises here, because what you start with is what you're going to get. But this does validate the framework that we used. The next two parameters in high tunnels were different, though. 14-day average temperature matters most, and the wind speed, whereas in open field, relative humidity actually matters more, and average temperature after that. So there is two different microclimates and microecosystems are happening here, and it's very cool that we are able to look at it all at once. So imagine 16 different parameters you're looking at and so many other variables all at once. And if you're just doing one by one, it would take you days or years. And that's the beauty of it. So long story short, machine learning is able to accurately forecast 88% thrips populations, parent populations, and temperature were the key predictors of severity, but there's also some micro variations within that. And then what this tells us that the field and high tunnel systems function as distinct pest microecosystems. And this is a very cool paper we recently published in Ecological Informatics. And if you're interested in the story and maybe how you can employ this in your systems, please do check this out. Where we are going next with this is we want to first improve the data quality. Now, what we use is a data from or weather data from a weather station that was within a 1 mile radius. But because the things are happening at the micro scale, the micro environment that thrips are experiencing, we cannot capture that quite yet. So what we want to do is to deploy in situ microclimate sensors that we're able to do that, so that will improve the model precision even further. Second thing we want to do is, this is all cool, but what does it mean for growers? So how do we tie the model results or population results into risk thresholds, something that growers can take and make the decision based off of? And that's what we are working on next. And then because TSWV is basically married to thrips, we want to tie the pest population dynamics into disease risk and see if we can move from insect level surveillance to the system level surveillance with the insect, the disease, and the environment, which is really important, and abiotic stress is built into that. So this is where all of this goes. What we try to do is to use RNAi as a potential next generation pest management strategy, improve thrips surveillance as well as tomato spotted wilt virus surveillance using machine learning, be able to better detect and efficiently detect tomato spotted wilt virus or discriminate potentially between strains using Cas13a based system, obviously classical ecological pest management, understanding molecular interactions between resistant host, virus, insect vector, and then trying to come up with the resistant cultivars that are more sustainable, that are able to withstand the tremendous pressure from tomato spotted wilt virus better. And all of this feeds into a system level approach where prediction is not the endpoint, but it's the connector. I would like to thank our collaborators, funding agencies, and this particular work that I presented here was funded through FFAR New Innovator Award, Texas Mag Research, and Texas Department of Agriculture. And if you step back, what we try to do here is pretty straightforward. Across this work, we have taken a different approach. First, to understand these strains at the molecular level, then detect them using Cas13a-based system efficient detection, using RNAi as a next-generation pest management tool, and then use machine learning to understand vector dynamics better in different microclimates. So what begins to emerge is not a single solution, but it's a framework. And it's a framework that allows us to understand, to detect, to control, and predict the system as a whole. And because if the pathosystems are dynamic, which they are, our strategies cannot be static. And we don't need the better control, we need the anticipatory control. And that shift from reacting to predicting is where the field is headed. I'll stop there and take any questions you may have. Thank you.
Questions, comments?
Yeah.
No, you can go first.
Very nice work. Just wondering the host range for the citrus virus you used for Vegas. Does it have a wide host range, or just think about the approach you developed for controlling other virus induced diseases?
That's a fantastic question. Yes, it has a broad host range. Sorry, I didn't go into details on that. Even though it started in citrus, obviously we are using it in tomato and pepper, and we know it goes in tomato and pepper. We are using it in Nicotiana benthamiana. We know it goes there. We know it goes in other vegetables and other crops as well, because it's such a small RNA.
Yeah, very small.
Yeah.
3 kb, right?
Yeah. And so I think the molecular finding with this was this RNA actually hijacks the phloem protein PP2 in cucumber. They did a bunch of studies, and this goes in cucurbitaceae as well. And that's how it moves from one cell to another systemically. And that's what makes it a great Wix vector, is that it's able to, once we figure out how to commercialize this, we'll be able to go in a bunch of different crops. Yeah. Yeah.
So my understanding with the system, you're just putting the target sequence into the, how stable is it? Is it work better than other viruses? Because sometimes when we're working with different viruses, we lose the inserts.
Yeah, no, that's a great question. So I think I have a slide somewhere which shows the CYVV, in the constructs that we have targeting both thrips as well as tomato spotted wilt virus, pretty stable till at least 50 days, we know, because a lot of vectors that we worked with in the past is like, yeah, you get the great system working for 10 days, but not for 50 days, or, that defeats the purpose. But this is more stable. Not that it does not have issues. It does have issues with something like Nicotiana benthamiana, for instance. When we started this work, we had a lot of plants dying, and we couldn't quite figure out what's happening. One of the things is that our postdoc infected plants they were too small. So we had to get them to like 2 weeks or so, or 3 weeks or so that they will be able to withstand the virus pressure. But yeah, that was one-time problem, and we have done several experiments since, and we never had any issue with that stability. Okay.
You had mentioned that you had used for tomato varieties the M82 and the Mountain Merit, which are both determinate varieties. Would you expect to see any significant differences in an indeterminate variety?
No, that's a great question. I think so, because these are the two of the cultures, and I do not know definitely if that's a definite yes, because we actually do use a lot of different varieties. These are just 2 examples, and we used them recently. We have a breeder who we work with, and he's based out of College Station. We have several different lines that he's developed, not just commercial cultivars, but several different lines that are in development against Tomato spotted wilt virus, screened against thrips. And this is the pattern that we are able to see, is that the resistance breaking strains in general do not care, at least the ones that we have, whether like what kind of cultivar it is. And when we reported this the first time, we actually tested that isolate from 2023 that I mentioned in 8 different cultivars. I don't know exact sources. Some of them were commercial, some of these are lines. Again, now we are using in the 26 field season completely different cultivar, because we are incorporating the new cultivars as they are getting developed or in the pipeline of development. Right now we are working with 4 different cultivars. 2 of them are resistant SW5, SW1. In one, we have one that is tolerant and one that is like susceptible as a kind of control. But I believe some of those is got to be indeterminate as well as determinate. Yeah. Good question. Yeah.
I was really excited about your Cas9 or your Cas13 work. That's really great. A question though is RNA extraction is still not field friendly. So do you have any ideas on how to deal with that part?
Yeah. So
we're like, how do we actually do the field?
Yeah, we don't want to obviously take the RNA to the lab and come back and do the rest. So what works with this is a crude RNA extraction. So it doesn't have to be like high quality RNA like you use for lab applications, right? So it could be just crude in lysis buffer. That's something we have tried, and it takes 10 minutes to do it. Now, there is an incubation step, and we would still probably need to figure out, and we are still fine-tuning this. Our goal is to ultimately be really field ready. And we are also trying to bring the time down from 40 minutes at the moment to something like 20 minutes realistically. So you don't have to wait for that long. But that's a good question. Yeah.
And then I didn't quite understand, are you just purifying the enzymes, or are you and mixing them?
These are commercially available for the most part.
Oh, that's good though. So
yeah,
anyone
yeah.
can use it? Okay.
Yeah, and then the guide RNA, obviously, you would need to synthesize it before, and it will become a part of that mixture. But yeah, it's all these are standard. Like the fluorophore is standard, the lysis buffer is standard, and the Cas13 would be standard. So you can just mix and.
Later we'll talk more.
Yeah, cool.
That's great. What is it about the green beans and the instar stages? Why are the green beans so suitable for that purpose?
Oh, because they're easy to work with. What we have is a great system. We do actually have sometimes issues when we do not get high quality beans for thrips rating, and we had people going from grocery store to grocery store, spending all afternoon to find the right kind of beans. So they have to be fresh, tender, because beans the thrips otherwise would not feed on them or lay as many eggs. What is great about this system is that when they lay eggs, they can lay in the green beans, and you can take them out. And what we like to do in our colony is that we like to maintain different instars, larvae, pupa, separate them out stage-wise. First instar larva, second instar larva, pre-pupa, pupa, and adult. You will find 5 different stages at any given time point. So it's great for moving them from one to the other. When the thrips lay eggs, for instance, right, you can just take the beans, put them in a new container, and then you can start with the first instar larva and then just move them across. So they're great for the containers that we use for rearing them. It's really the easiest system to work with. But this is something else, actually. There's a paper that recently I looked at like a couple of months ago. They've been using soybean, something that seems to be even easier. That's something we're going to try. It's a part of soybean. I'm not remembering what it's called, but you can easily grow that in the grow chambers, put the thrips on, and it's apparently really easy to move them across stages as well. But yeah, that's the easiest system to work with.
A couple questions from online, and I think that'll wrap us up. So Catherine asks, hi, Dr. Gadhave, I really enjoyed your talk. I'm curious about the more sensitive microclimate sensors you mentioned in the machine learning project. What can these detect that traditional weather sensors cannot?
Oh, that's a great question. So the goal with that is we have high tunnels, we have field. So what we set up just last week is the weather station within one high tunnel, which is about this size, right in the center. So we are able to detect, and these are, conventional. These are not any special, but what we want to get is at least 4 different weather parameters. We want to get the temperature, humidity, humidity, wind speed, and wind direction in high tunnels, and those are the ones that matter the most, which you cannot put the hobos and get that data on wind. So that's why we wanted to establish the local weather station within the high tunnel, and that would be the case with the other high tunnels. So each high tunnel will basically have its own local weather station, which is recording data in high resolution. We can go minute by minute even, or every 15 minutes being recorded over the entire growing season and more, because we are establishing those traps 2 weeks before planting and 2 weeks after. So we wanted to get that activity and then see how that correlates with the microclimate within the high tunnel, whether that would vary between one high tunnel to another, or that would vary. Obviously, we know that it varies between high tunnels and open fields. And so we will have a local weather station in the field itself as well, and compare what that does to the thrips populations. And that's where we are headed next.
Great. And then Steph says, such exciting work. Are there any data showing how early in infection the Cas13a detection method can detect TSWV in plants? For example, 1 day after thrips transmission, 1 week, etc.
Oh, that's a fantastic question. We do not actually know at this point, because we did not do the time course analysis as to how early can you detect it. But that's something we'll work on in the future. Because we are able to, with the TSW, it's because it's such an aggressive virus. Even in early stages, you got to be able to have hundreds or thousands of copies even early in the infection. And I think we should be able to detect those as early as it could be, but I do not know that for sure unless we do those experiments. But great question.
All right. Yeah, that's fantastic work. So let's.
Thank you so much. Thank you.